I'm Alex Turner, and today I'm going to talk about some work on extreme events driving year-to-year differences in gross primary productivity across the United States. I thought I'd start with a very brief explanation of why I began looking at this. So I was working on urban CO2 in the San Francisco Bay Area, and there's quite a bit of complexity in this region with a mix of both anthropogenic and biospheric activity. I was struggling with how to constrain the CO2 fluxes from the biosphere, and around this time is when Philip Kohler's GRL paper on TROPOMI SIF came out. I was pretty excited by this paper, and others, so I decided to look at what it could tell us about carbon fluxes in an urban area. So I actually went ahead and just plotted up the data over my domain, and the figure on the right is what I got. For reference, this purple star is the Moscone Center, where we'd be sitting in a normal year. So the general patterns kind of match up with what you might expect for the Bay Area, with more SIF over the East Bay Hills to the east of Moscone Center, and up on the peninsula in Marin County north of San Francisco. But to be honest, it was a little bit disheartening because it wasn't clear that this was sufficiently high resolution to address the problem I was interested in. But before moving on, I decided to try something else out that started me on a fun project looking at the biosphere. I'm an atmospheric chemist by training, so I decided to use something that's pretty common in that area, which is called oversampling. The basic idea is that the actual footprint observed from space varies over the orbit cycle. With TROPOMI, we get about 8 successful SIF retrievals every 14 days. So we can use those different viewing geometries to obtain higher spatial resolution than the actual satellite instrument. With TROPOMI, the native resolution is about 4 kilometers by 7 kilometers. It's actually finer in the long track now due to reduced integration time. And this is what a hypothetical swath might look like. And here's what a second swath might look like overlaid on top. And a third, fourth, fifth, sixth, seventh, and eighth. So you can see how the different pixels bisect each other, and that might allow us to obtain higher spatial resolution than the actual satellite footprint. The assumption we're making here is that the underlying signal is varying slowly enough that we can use measurements over multiple days to constrain that signal. As such, we're going to be building a 14-day moving average that has higher spatial resolution than the native TROPOMI footprint. But there's actually an additional thing we can do. And so let's focus on a single scene for a moment. Let's pretend it's this gray pixel. There's additional information we can leverage, and what I mean by this is we know that the fluorescence we observe from space originated from vegetated areas. And this image on the right shows the actual spatial footprint for a single scene. What we can see is that this particular scene has a body of water bisecting it. So any fluorescence we observe from this scene should have come from areas of vegetation, not the water. So what we can do is we can redistribute those photons, or that SIF, based on the underlying vegetation distribution that we get from a higher resolution product. So we can use this essentially as a subgrid weighting and redistribute the SIF within the scene. And the nice part of this is that it redistributes the fluorescence to where it's likely emitted while conserving the photons over a given scene. We can then do this subgrid weighting for each satellite scene and put it all together. So here's what we started with. This is what oversampling gives us, which is essentially a smoothed version of the original data with maybe a little bit higher resolution. And then here's what downscaling gives us. I was admittedly pretty excited by this because we're actually seeing spatial patterns in SIF at the urban scale. And for reference, this is where Berkeley is. Yeah, go Bears. And this green patch here on the left is Golden Gate Park, which would be a little bit to the west of Moscone Center. And then it's a little bit hard to make out here, but this faint line between Cal and whatever it is, Highway 24, is actually Highway 24 that cuts through the East Bay Hills. So it seems like we're seeing SIF at these urban scales, which I find pretty exciting. All right, there's still the outstanding question though. How does TROPOMI SIF actually relate to GPP? And so what I'm showing here on the left is GPP estimated for the Bolton Island corn site in California's Central Valley. The gray dots are measurements over the course of the day, and the black dots are measurements between 1 and 2 p.m., coincident with the TROPOMI overpass. And then the green dots here are the TROPOMI SIF observations at this site at the same time. So we see a strong agreement between the two, with SIF seeming to capture both the spring and summer peaks in 2018 and 2019. And plotting them against each other, we see that SIF explains about 79% of the variability in the Ameriflux GPP data. And personally, I was pretty excited by this because we never see this kind of agreement in atmospheric science. So I thought it was pretty exciting. We also looked at other ecosystems. For example, the middle panel shows data for the Vira Ranch site, and the right shows the Tonzi Ranch site. The middle is a grassland, and the right is a savanna site. Both of these are in California and operated by Dennis Baldacchi's group at UC Berkeley. In both cases, we see that SIF seems to capture the temporal evolution of the GPP data from the Ameriflux, albeit with a fair amount of noise. But overall, we're seeing a fairly strong correspondence between the SIF data and the GPP data across ecosystems. However, our California analysis only covered a handful of ecosystems. So we've now extended to all of CONUS to include 82 Ameriflux sites, shown here as the yellow stars, that have data in 2018, 2019, or 2020. And we also show some representative ecosystems on the map as well. So if we plot all of the SIF data against the Ameriflux GPP data, so SIF on the x-axis here and GPP data on the y, we find, again, fairly good agreement with a reasonably linear slope. What we still wanted to know is how many distinct SIF-GPP relationships are there in the data set. So what we did was we did a bootstrap analysis for each ecosystem, where we generated 2,000 realizations of that SIF-GPP slope for a given ecosystem, and then plotted them as a histogram, which can be seen on the left. So each color indicates a different ecosystem. Then what we did was we fit these distributions with a Gaussian mixture model, and we found that a mixture of two Gaussians best explain the data. You can see one on the left in the middle of the grassland, or the blue distribution, and then one sort of in the middle centered around 16.4 or so, which covers most of the others. So what we're finding is that only grasslands stand out as distinct, and the rest seem to have a fairly universal SIF-GPP relationship. We're still investigating why the grasslands seem to be distinct. We suspect it might be related to canopy structure. But overall, we're observing two distinct SIF-GPP relationships across ecosystems. So we can then put this all together, and what I'm showing here is an animation over all of CONUS. So the bottom left shows a little clock, so it shows year and month, and the animation is going to show the estimated GPP data from TROPOMI SIF. And I find this pretty exciting because we can start seeing pretty interesting spatial patterns, although now we've gotten to sort of the winter time when it gets a little bit more dormant. What you'll end up seeing is that California and the Central Valley on the left light up first, and then what you'll end up seeing after that is the eastern US begins to light up. So here California and the Central Valley begins to appear. You can also see the Imperial Valley down near San Diego. Now the eastern US is beginning to wake up. You can see the Willamette Valley up in Oregon. The Mississippi Alluvial Valley is pretty distinguished. And then come summertime, you see the Corn Belt and all of the Midwest begin to appear. And then again in winter, things start to go dormant. All right, so we can also look at the differences between the years. So what I'm showing here is data from 2019 minus 2018, so showing the year-to-year differences. And a few patterns stand out pretty strikingly. For example, we see this patch in California jump out, which I'll talk about in a moment. And we also see patches jump out in Texas and the Midwest, which again I'll talk about later. So what I'm showing here in the bottom left is the data across California, so the GPP inferred from TROPOMI as the black line. And what I can do is I can overlay the 2018 seasonal cycle onto the 2019 seasonal cycle. So we see the main difference between these 2 years really seems to occur in fall. And what I'm showing here on the right is the data just from fall. So we see that across California there seems to be a fairly uniform difference, with possibly a stronger signal up in Northern California. So it seems to be a statewide signal that we're seeing. We can also overlay the precipitation data shown in blue. So this is the cumulative precipitation over the water year. And what you'll notice is that in 2019 we get about twice the amount of precipitation as we did in 2018. The other thing you'll notice is that a lot of the rises in SIF or in GPP correspond very strongly with precipitation. Particularly you can see this in 2018 right around mid-March, where you see a big jump in both GPP and precipitation. And this is in part because in California a lot of our water comes in the form of atmospheric rivers, or otherwise known as Pineapple Express, sort of bringing water up from the tropics. And actually what I'm showing in the bottom right here is an animation, or is an image from Goes West showing one of these atmospheric rivers. Actually, this one from March 2018. And you can see that a band of precipitation just comes up from the tropics and really covers all of California, leading to sort of a statewide greening. And so actually between these 2 years, 2018 and 2019, one of the big differences is that in 2018 we only had really 2 major atmospheric rivers, whereas in 2019 we had a series of big precipitation events early in the season that ended up giving us almost twice the amount of precipitation. So it seems like this factor of 2 increase in precipitation in 2019 served to extend that fall growing season relative to 2018. However, interestingly, if we overlay data from MODIS, for example, NIRV, we don't really see much of a difference between the 2 years. And this is actually kind of interesting because if you go back to some work from Sellers in 1985, he argues that greenness indices provide estimates of photosynthetic rate provided optimal soil moisture, temperature, etc., and PAR is known. So what this might suggest is that what we're seeing is a downregulation of photosynthesis in fall 2018 due to a water limitation in the TROPOMI SIF data, or here plotted as TROPOMI GPP, that's not present in data from MODIS because we don't have those sort of optimal soil moisture conditions. So we think we're seeing a downregulation of that photosynthesis in response to a water limitation, which I think is kind of exciting. So moving on to some other large anomalies that we see, what I'm showing here is the anomaly in Texas, and we can do a similar analysis. So again, what I'm showing is black is a TROPOMI SIF inferred GPP, or sorry, GPP inferred from TROPOMI SIF. Blue is again precipitation, cumulative over the water year, and then green is again an IRV from MODIS. However, for this case, what we see is actually, well, there's a couple things that stand out. First, again, we see a lot more precipitation occurring in 2019 than in 2018, and this leads to a dramatically different seasonal cycle in both NIRV and in the GPP inferred from SIF, with 2019 having a much larger springtime GPP and NIRV signal. But interestingly, actually, both SIF and NIRV seem to show similar seasonality in this Texas case. So both of them see this drought suppression of GPP in 2018. Although somewhat interestingly, you actually do see a secondary peak in 2018, which is driven by some late precipitation that occurred around August, giving us this peak in GPP in roughly September, October. Another major anomaly I wanted to point out is the big blue patch here in the Midwest. This is across the Midwest Corn Belt in Illinois, Indiana, and Ohio. And this is actually something that was highlighted by work from Yi Yin in AGU Advances this year. But really what happened here was again driven by precipitation, but there was a flooding in 2019 that delayed the onset of the growing season in the Midwest. And actually you can see that delayed onset in the bottom right in 2019. And it's again present both SIF and NIRV here. So overall, we're seeing that GPP across CONUS varied by less than 4% between 2018 and 2019. But most of the big deviations that did occur were really tied to precipitation anomalies. So overall, this work was focusing on extreme events that are driving these year-to-year differences in GPP across the US. And so what we did in this work is developed an ecosystem-specific GPP estimate from SIF at 500 meters across CONUS. It's a daily estimate that is representative of a 2-week moving average. We find that United States GPP differs by less than 4% between 2019 and 2018. But those differences that we do see are primarily driven by 4 major precipitation anomalies that account for 28% of the year-to-year differences. And the work that I showed here, part of it's published, the early part on the downscaling and oversampling was published earlier this year in Biogeosciences. That work was titled A Double Peak in the Seasonality of California's Photosynthesis as Observed from Space. And the other work is currently under review, but I'd be happy to send a preprint along if anyone's interested. And feel free to contact me at my email address there, turneraj@uw.edu. Thanks for listening.